Welcome to The Explainer. Today, we're going to dive deep into something pretty wild. We're looking at how scientists are engineering failure in a petri dish, all in the hope of finding a cure for muscle disease. So how can you safely test cures for muscle disease in a petri dish? I mean, this is a huge challenge in medicine right now. Millions of people suffer from muscle-wasting diseases, and studying them is just incredibly hard. The solution, and this is the really cool part, turns out to be not about figuring out how muscles grow, but engineering them to fail right there in the lab. Okay, so before we get to the solution, we've got to wrap our heads around the problem itself, right? We're talking about muscle atrophy, which is just the scientific term for muscle wasting. And if you want to study it, you need a way to copy what's happening inside the body when muscle starts to break down. You know, healthy muscle is just amazing. It all starts with these tiny little cells called myoblasts. The best way to think of them is like Lego bricks. A single brick isn't much, but when they get the right signal, they start clicking together. They fuse, they share their internal parts, their nuclei, and they build these long, powerful fibers called myotubes. And this fusion process, it's everything. It's how our muscles grow and how they repair themselves. So what happens when that building process just stops? Well, that's muscle atrophy. Whether it's from an injury, a disease, or just getting older, those little myoblast cells just stop fusing. The muscle can't build new fibers, it can't repair the old ones, and the result is that weakening and shrinking that is the hallmark of atrophy. Okay, so if you're a researcher and you want to study this, you can't just use a standard plastic petri dish, no way. You need to build something special. You need to engineer an environment that can actually make muscle atrophy happen right when you want it to. And their solution was brilliant, a custom-designed hydrogel. You can basically think of it as a really sophisticated jello for growing cells. And they made it in 3 main steps. First, they start with regular gelatin. Then, they chemically modify it to create this gel base called gelma. And then, and this is the absolute key, they mix in a special polymer called PSS before the whole thing sets. Okay, this diagram looks complex, but the secret sauce is right there, that PSS polymer. What's so special about it? Well, it's designed to mimic a molecule that's naturally in our bodies, and it gives the surface of the hydrogel a really strong negative electrical charge. And believe it or not, this tiny shift in electricity, well, it changes everything. And this is what makes the experiment so clever. The researchers made 2 different hydrogels. Physically, they were identical, same stiffness, same structure, the whole deal. The only real difference was that electrical charge. The standard Gelma gel was a tiny bit negative, about minus 1.3 millivolts. But the special Gelma PSS gel, it was way more negative, clocking in at around minus 9.5. That's a huge difference. All right, so the stage is set. The team takes these healthy muscle cells and puts them on 3 different surfaces. Their special negatively charged gel, the standard gel for control, and just a normal plastic lab dish. The expectation? They figured they'd see healthy, happy muscle fibers growing on all 3. But that is not what happened. Not even close. On the standard surfaces, yep, everything looked great. The cells fused together perfectly, creating these beautiful, long muscle fibers. But on the special Gelma PSS surface, wow, it was a total failure. The cells were alive, they were growing, but they would not fuse. They just stayed as these little individual undeveloped cells. I mean, just as the researchers themselves said, the platform was actively preventing the cells from doing their job. That negative charge was somehow putting up a giant stop sign, blocking the whole fusion process. They had done it. They had created a perfect little world for muscle atrophy, right there in a dish. And the numbers just back this up completely. They used something called a fusion index to measure how many mature fibers actually formed. And get this, on the control surfaces, after just 3 days, about 80% of the fibers were fully mature. And on the special Gelma PSS platform, a measly 3%. It was a near total shutdown of muscle development. Just incredible. So this was obviously a massive breakthrough. But it immediately led to the next, even bigger question. Why? What was it about that negatively charged surface that was telling these cells don't fuse on a molecular level? Well, they looked at the cell's genetic activity, and they found the smoking gun. That special surface was basically turning down the volume on all the most important genetic signals for muscle growth. The pathways that shout, grow, mature, and even grab on tight, all of them were being silenced. So here's what was really going on. The cells just couldn't get a good grip on that super negative surface. It's like trying to climb a slippery wall. And because of that weak connection, their internal machinery got all messed up. A specific protein called focal adhesion canine wasn't getting the right signals. And without that working properly, the main grow and mature commands were just lost in translation. The cells were basically in a state of confusion, waiting for instructions that never came. Okay, so this is all super interesting in a lab dish, right? But here's the million-dollar question. Does this man-made atrophy actually look and act like the real thing? Does it match what happens inside a living creature after a real muscle injury? Well, just look at this. The comparison is staggering. On the left, you've got the lab-grown atrophied cells, and on the right, that's a real sample from a mouse muscle 5 days after a serious injury. They look almost identical. You see the same chaos, the same lack of those nice, long fibers. The visual match is just undeniable. And it's not just about looks. The similarities go way deeper. Both the lab model and the real injury show that exact same problem with cell fusion. The same disorganized mess of fibers. And this is crucial. They both show that same shutdown of the critical grow and mature signals. So the platform wasn't just making unhealthy cells, it was perfectly recreating the exact biological fingerprint of real-world muscle atrophy. So, what does all this mean? What does having this incredible new tool actually do for the future of medicine, for people who are actually suffering from these conditions? Well, for starters, it gives researchers a reliable and, more importantly, a repeatable way to study muscle atrophy whenever they want. They don't have to depend entirely on complicated and slow animal testing for that initial research anymore. They can now just create atrophy in a dish, on demand. And this is the real game changer. This little platform is absolutely perfect for testing new drugs, and testing them fast. Scientists can now take thousands of different compounds and just throw them at these atrophied cells, looking for one that can break the spell. One that can override that don't fuse signal and get those cells to finally link up and build healthy muscle. This could just dramatically speed up the hunt for new treatments. It's really a fascinating idea, isn't it? By so brilliantly engineering failure, these scientists have opened up a brand new road towards success. And it really leaves you wondering, could a simple polymer hydrogel, this little patch of negatively charged gel, actually unlock the secrets to rebuilding our body's largest organ? It's an exciting thought. Thanks for joining this explainer.